Good afternoon and welcome to the Protection Island Speaker Series. Today I'm happy to introduce to you Dr. Andrew Cameron. Andrew will be presenting the COVID-19 pandemic, current topics in vaccination, public health, and the evolution of viral variants and their implications. Andrew graduated back in 2000 from VIU, then Malaspina, with a BSc in biology. It was as an undergrad there he spent a summer doing a field course and also an extended fellowship in Belize, where he discovered a love for microbiology. Next, Andrew completed a PhD in microbiology at UBC and a postdoctorate at Trinity College in Dublin, Ireland. Currently, Andrew is associate professor in the biology department at the University of Regina and is co-director of the Institute for Microbial Systems and Society at the University of Regina. Andrew works on diverse aspects of COVID, including leading multiple research projects, lecturing, and he leads the university's recovery planning group about the pandemic, vaccines, immunity, forecasting, and the virals. Also, Andrew works on the development of new genetic tools for viral detection and variant identification. He works in partnership with the BC, Alberta, Saskatchewan, and Manitoba Provincial Public Health Laboratories, as well as the National Microbiology Laboratory in Winnipeg. Andrew is married and currently resides on Protection Island. Many of you will know Andrew and also his dad, Norm Cameron, who has lived here for many years. Now, Emily will mute you during the talk, so please use the chat box if you have questions during the presentation or raise your hand. There will be a question and answer period at the conclusion. Welcome, Andrew, and over to you.
Thank you very much, Bev, for that introduction, and great to meet everyone. And some of you I know already, and I really look forward to meeting everyone else. And as Bev said, I am now a resident of Protection Island. I'm at 142 Pirates Lane, renting currently from a couple, Ryan and Rosie, that some of you may know. And so I look forward to either bumping into you if I ever leave this office. That's one I guess pitfall of being a COVID researcher, is that one very rarely gets to leave their computer these days, because there's always something happening. So great, thanks very much for this invitation. And as you heard from that introduction, I mean, this is a big topic, right? And there's so many different things to talk about when it comes to COVID. So I'm going to try to keep my presentation relatively short, so that we can get to conversation and discussions about any one of these topics and really let it sort of grow from there. Now, that said, I do have some slides to cover both an introduction of myself and general research, because this is a bit of a homecoming, and it's, again, a community that I'm looking forward to becoming more and more enmeshed in and a part of. And I'll tell you a little bit about the research that we're doing in COVID, but most of the focus will just be on sort of big COVID topics. So the issues of vaccination. What are variants? I'm going to start with what are variants, because that's just the most topical thing at the moment. And so hopefully I can help us make some sense of this massive information. These are really such unprecedented times in so many ways. And as a scientist, it's unprecedented because never before have we had an onslaught of information like we're really experiencing now. So whether that's all of us in our communities and everything we're seeing in the news, to then we scientists who are working on this, that science doesn't normally move at this pace. And there was a review recently that up until January, there was about 80,000 peer-reviewed publications on COVID that had come out in the past year. Now, unprecedented is the best word for that. That's just try to do the math. They're trying to read 80,000 papers on something. It's impossible, right? So I think that's my way of prefacing that this is a big topic. I want to discuss it. I certainly don't have all the answers, but I really encourage you to reach out to me if you'd like to follow up any of these discussions, because what I can do well is usually find answers and find the fastest ways to really trustworthy information and then help interpret what the science is finding these days. So with that, I'm going to start sharing my screen, and I do have my chat window open. So if you have a a burning point to make or question, then please fire it in the chat, and I may stop and address it in my presentation, or we will definitely have a question and answer at the end. So I'm going to share my screen. And hopefully you see my title slide, and it's a nice big full screen. Emily, Bev, look good?
Yes, it does look good.
Great. Okay. Right, so in my introduction, you heard that I have a long history here. So not quite born, but raised in Nanaimo. And I've really been privileged to return now to Vancouver Island University and start to build a new connection there as an adjunct faculty member. And hopefully that will again grow as we start to get through some of these pandemic times. But I do spend 200% of my time really online at the University of Regina these days, leading multiple COVID research programs and helping with COVID administration at the university. But more generally about me and as a scientist, that I'm really interested in really microorganisms everywhere and on every surface and in every little nook and cranny. Now, often my research tends towards pathogens, but although I study a lot of infectious diseases, I would say I often apply this One Health framework that is a term that's becoming quite popular now to express that to really understand infectious diseases, we need to understand the environments and the animals and the humans around us, that everything is really connected in the transmission of diseases. And we see that so clearly with our current COVID pandemic. SARS-CoV-2, as you may know, is a virus very recently derived from a bat, and it jumped into a human. So part of our research is interested in trying to figure out, how does that happen? How often can that happen? And what set up the conditions for that to happen? So there's me walking awkwardly across native prairie. So for the past 9 years, I've been living in southern Saskatchewan before relocating here to the West Coast just before Christmas. And I have various Protection Island connections. In 2016, I was very fortunate and privileged to travel with the Village Medical Project for Sierra Leone. You can see Wes sort of grainy in the right-hand side there, to go work in the Bonabou region and chiefdom with Wes and I was still, when I was getting started with some of these genetic technologies I'll tell you a little bit about, to see what types of samples we could take and what types of discoveries we could make about infectious diseases in many, many different environments, and particularly antimicrobial resistance. So that's one of my big interests, which I won't have a chance to talk about today. And since then, I've started to do a lot of work in Southern Africa. And there's a picture of Botswana, where I've been spending a fair amount of time over the past 3 years, working on the connections between that river there, the Chobe River, and the massive amount of wildlife that lives on that river, and then the connections to the few human habitations along that river, and looking at how diseases cycle between all those different landscapes and animals and humans. And then, of course, in Saskatchewan, have to do a little bit of agriculture research. And so we now have some pretty exciting projects going, looking at the evolution of antimicrobial resistance in farm dugouts in Saskatchewan and connections to our food chain and humans. And also I work on various coastal ecosystems as well. So for example, one project at the moment with the BC Centre for Disease Control, we're working on using our genetic technologies that we're developing in Regina to understand tick-borne diseases better. So things like Lyme disease and other infections that can be spread by ticks. But as I said, I'm a microbial geneticist, and most of my work is very much on the molecular level. And to understand microbes and pathogens, there's many different ways that we can use genetics as a tool to really interrogate these different systems. And here's a broad overview of the types of approaches that my lab takes. So in addition to the molecular epidemiology in the upper left-hand corner, which is part of what I'll tell you about today, we do other types of studies as well. So we study little defined ecosystems of microorganisms. We also study broader ecosystems, going out and sampling soils and trying to understand the thousands of different bacterial and fungal species that live there. We do a lot of genetic manipulation, where when we think we might have tracked down a gene that might confer antibiotic resistance in a pathogen, then we'll go into the lab and we'll tinker with that gene to see how does it confer resistance. So that's what you see in the bottom left-hand corner, where we figure out how do organisms evolve resistance to new antibiotics. And then we're also very interested in how genes move through environments. So how do genes pass from one species to another? Because as you may know, with things like antimicrobial resistance and pathogenesis, bacteria and viruses, they're always moving genes around, that genes can jump. And that jumping of genes is very important for understanding where do pathogens come from and how do they evolve. But as I said, today I'm going to focus on that molecular epidemiology. So that upper left-hand side there is showing you that colorful pinwheel on the left-hand side is a phylogeny, and that's taking about 4,000 SARS-CoV-2, so the virus that's causing COVID, comparing their genomes and then looking at how the virus is diversifying as it spreads around the world. And then you can see that same color coding applied on the right-hand side to look at the diversity now of SARS-CoV-2 viruses in different countries around the world. And I need to introduce all the fantastic people I work with, or most of them are captured in this photo from pre-COVID times. This Institute for Microbial Systems and Society, I co-founded it along with Chris Yost, who you see in that photo there, back in early 2017. And since then, we've been growing to work on diverse projects across agriculture and industries. And my realm is really infectious disease. And here I'm going to highlight a few of the people whose work I'm will be telling you a bit about today. I'm not going to present much data, but I'm going to tell you a bit about how we're going about understanding SARS-CoV-2. So highlighted right in the middle on the biggest yellow circle there is Dr. Kara Luce, and she's a genomics research associate who really runs my physical lab space. That's what allows me to get to work in an office here in Saskatchewan. And she's directing our COVID projects at the moment. And in pink there, you see Dr. Keith McKenzie, who was really co-leading the COVID projects with Kara up until about October, when the public health lab came knocking and recruited him away, so that now Keith does all the genome sequencing to find variants of SARS-CoV-2 in Saskatchewan. So that's another great success story. We keep these great connections because our trainees from our institute are being recruited very quickly by the public health lab. And in fact, 3 people in that picture there now either are straddling between our 2 labs or are now working fully in public health to try to address this COVID emergency. And in orange there, Kevin Kaczynski is at the BC CDC, and it's a technology that he's been pioneering that I'm gonna come around to tell you about at the end, that we're applying to understand not just SARS-CoV-2, but all the other viruses and bacteria that cause infections in humans that we just don't know about. And that's what's really exciting about genomics and using genetics, is that we can discover so many new things that to this point we've just been blind to. We didn't really know were causing disease and circulating in our communities. Okay, so let's talk about COVID. We're in a very interesting time. I would say this is, I don't know what adjectives to apply to the past week, but it's definitely a new phase in the pandemic. And we are watching as infection rates are rising. You can see in Canada there, in that past 2 weeks, really the past week highlighted in that gray bar, how infection rates are going up. And we had such positive trend there through January and February of infection rates declining, but now we're bouncing back, and some are referring to this as a potential 3rd wave now. You can see that we never even got down to very low levels really across Canada though. And then specifically in British Columbia, again, there's now this increasing trend upwards of more and more infections, when we heard one of the highest numbers yet, I think was it on Friday, of new infections in British Columbia. Worryingly, hospitalization rates are climbing in some areas as well. And this may well be due to these variants of concern, which I'm going to introduce in the next couple of slides. Okay, so yes, the variants. What are they and where do they come from? I'm working a little bit backwards here to how I would normally introduce COVID, but I think we should just jump right in and talk about the variants because we're hearing so much about them. So on the left-hand side there, you see a person who I've illustrated exhaling COVID, the SARS-CoV-2 virus is all the little red dots. And someone in their local area, if they encounter a few of those viruses or too many are inhaled, then that person can become infected by the virus. And so this is the transmission that's now resulting in millions of infections around the world. And the bulk of that transmission is by droplets. So the closer you are to an infected individual, of course, the concentration of virus that they're exhaling is going to be highest. And most of that transmission happens in those close quarters when people spend extended times together. It can also transmit by aerosol, which means small amounts of the virus can float in the air for longer, but that doesn't explain as many infections as does the droplet transmission. There's also the potential to pick it up from surfaces, but it appears that that's a very minor contributor to the current pandemic. Now, what has just changed in there, you might not be able to see it, but one of those viruses inside that person's lung, or what really is their upper respiratory tract, where most of the virus is going to be replicating, has just changed and it's mutated. And that's me illustrating that variant. And what's happening with variants, and I don't want to get too down into the weeds here, but as a biologist, it sort of pains me to use the term variant because we betray the fact that this virus is mutating constantly. And I'm going to illustrate that here. So I'm pleased now that we're turning to using these variants of concerns, and we're just going to talk about certain variants that the media now has adopted this term. So as I said, we know that SARS-CoV-2 came from bats, and the genetics is very clear on that. It may have passed via another mammal for a short period of time before it jumped to us, but definitely bats are the global source of the coronaviruses that do jump into humans at different times. And that little red dot there, so that's me just illustrating that very first virus. If we were to pull it out of the very first person infected, which was a person undoubtedly somewhere in southern China, then we could sequence it and figure out how it is then related to all the viruses that have come since as it has spread and diversified around the world. So that was somewhere in maybe October 2019 that first infection of a human happened. And now if we look at the number of mutations that have been accumulating as the virus spreads, this is coming from that genome sequencing, so taking all the genetic material out of the viruses and sequencing it. And these dots are colored according to different countries around the world. And you can see that as the virus is transmitting over time, it is accumulating mutations. And in fact, we don't even see the old original virus sequence anymore. It's probably gone extinct. So everything that's circulating now is technically a variant because the virus just continues to mutate and diversify as it spreads. Now, most of these mutations that are illustrated there are irrelevant. They don't mean anything to the pandemic. They don't change probably how the virus is evolving or able to interact with us. But some of those mutations are very, very important because they are changing how the virus interacts with us. And so an important thing to remember is that this virus was up until late 2019 evolved to live in a bat or another mammal. And in jumping to us, it wasn't a perfect fit to infect us. And so what's happening as it's spreading and diversifying is it's now experimenting with different mutations to find ones that help it use us as a better host. And that's what we're watching now. And most of our attention is turned to the spike protein. So there's a picture of the SARS-CoV-2 virus, and those red blobs on the outside are the spike protein. And why we're so interested in that protein is really twofold. One is that it's the protein that allows the virus to bind to our tissues. So if it didn't have spike, it couldn't bind to us and it couldn't cause an infection. So that's what mediates that interaction and allows it to infect us. It's also the primary target for our immune system. So if our immune system can target those red proteins and do things like make antibodies against it, then that's going to be very effective at preventing the infection. So all of the vaccines that are currently being deployed and are in development, all are focused on that spike protein. That's the basis of our vaccine technology now. So if we zoom in on one of those spike proteins, and what I'm doing here is just stretching out the protein to look at its sequence, and all these little peaks we see are different mutations that have occurred in that protein as the virus has been spreading around the world. Now, as I said, most of those are irrelevant to us. I mean, they're interesting to me as a bacterial geneticist, but they don't really mean much for the pandemic. They do help us track the virus, so that's pretty cool. But in terms of the virus adapting to use us as a better host, there's two mutations that we're hearing about in the news all the time now. These are the ones of particular concern, and that's hence the term variants of concern. And they are the E484K and the N501Y. And I've highlighted the two positions there. In the South Africa and then the Brazil variants, and now there's a new Philippine variant, each of those has evolved independently, but each one has acquired those two mutations. And those two mutations are very important because we think that the N501Y improves the affinity of the virus for our tissues so it can bind better. And I'm going to show you the consequences of that in the next slide or two. And the E484K appears to help the virus escape or evade our immune system. So these are two mutations that are very important because one helps the virus bind better and the other helps it evade the immune system. And there's actually some evidence coming out this past week that maybe the N501Y also helps the virus evade our immune system. So that's why we're particularly concerned about these. The England variant, the one that's spreading the most widely in Canada at the moment, has just that N501Y mutation. But that's very significant because we can see that these variants of concern compared to what we'll now start to see called in the news the wild type, so that's just the old-fashioned form of SARS-CoV-2, which is still the dominant form around the world, that these variants of concern are more transmissible, and I'll illustrate that in just a second, and they're also more dangerous. So they can cause a worse disease, which means that they can make younger people sicker, and they're also resulting in a higher incidence of mortality. So they're causing more death. So what's happening is that because the virus is adapting to use our tissues better, it's able to replicate to higher numbers. So when a person is exhaling, they don't need to be coughing or sneezing, just me talking right now, I'm exhaling little droplets that we can't see, but they are there, that there's going to be more virus exhaled by a person who's infected with one of these variants. Now, that's bad because, of course, you can see how that's going to help transmissibility if that person's ejecting more virus. The other really bad part is that it takes the recipient, the person who's become infected there in that illustration, they had to inhale fewer viruses. Because the virus is better able to bind to that new host, a person it's a lower infectious dose, so fewer viruses need to be inhaled to cause that infection. So for the next few slides, I'm going to talk about how we stop this. And one thing that's important to bear in mind is that these variants of concern are still stopped in the same way. So distancing still is a key tool in preventing transmission. Wearing a mask works brilliantly to greatly reduce transmission of the variant or the wild type. And then vaccines are going to be our primary tool to really stop this pandemic. And the race is on. And at the moment, we're not winning the race, but we need to get vaccination rates higher because that's the only way we're going to stop COVID altogether and stop the spread of these variants and, importantly, the emergence of new variants. Because as long as we let the virus continue to spread, it's going to keep evolving and new variants are going to arise. So let's talk about vaccination and the benefits of it. Just so there's a question there, are the variants of concern still the same symptoms? It appears that yes, so the symptoms are the same. We still have a pretty poor handle on when we have no way of predicting if a person's going to become symptomatic. So some people do suffer symptoms from the infection and others don't. And what's really problematic is that many young people don't suffer symptoms, but they're infected and they're transmitting high levels of the virus. So it's a bad disease in that way, that it's a bit sneakier than some others that we're used to. Okay, so let's talk about vaccines. So an unvaccinated individual on the left-hand side there, what is very, very clear, and we have tons of data now to support this, is that an unvaccinated individual is going to be much more susceptible to the disease. They're both going to be more susceptible to contract it, and they're much more likely to suffer severe disease symptoms and potentially death. And they're probably more susceptible to reinfection. So if they recover from the virus, they're probably more susceptible to contracting it again, as opposed to a person who's been vaccinated. So those are some huge benefits to vaccination, is that a person is going to be much, much less susceptible to disease, and they're going to be very much less susceptible to reinfection 6 months or a year, 2 years down the road. That's still a prediction. We don't have the data to confirm that yet, but based on what we know from vaccination against other infectious diseases, this is a very strong prediction at this point. Okay, so an unvaccinated person is going to shed more virus. So they're the perfect host for the virus. If they're unvaccinated, their immune system's never seen it before. So the virus is going to get in, replicate to high numbers, and a person is going to exhale, cough, and sneeze out a bunch of that virus. A vaccinated person is probably still going to be a host for the virus, but at a greatly, greatly, greatly reduced level. So this is because vaccines don't actually sterilize, right? They don't completely obliterate the virus. They don't completely prevent a person from being a susceptible host. What they do is, and we're seeing this with the current COVID vaccines, is that they do prevent the symptoms and the disease and the death, but there's a good chance that even a vaccinated individual is still going to be able to carry the virus. Now, they're also, even if they can carry it, they're going to be much, much less risk to those around them, because the virus is going to be very constrained in how much it can replicate. So that's why I've illustrated that person exhaling just a small amount of the virus. Now, if we put masks into the picture, right? So a person who's vaccinated and wearing a mask, then they are very, very, very low risk, because they're harboring so little virus, and what little bit they are exhaling is very likely to be caught in that mask. And we know that masks make over a 10 to 15-fold reduction in the amount of virus that a person is exhaling. And that is the difference between allowing the outbreak to persist and grow versus stopping it. So masks are still going to be important until we drive this virus extinct. Fingers crossed. And then another key point, and I hope this is something that starts to change in our culture, is that people stay home more when there's a risk of infection. So whether you're symptomatic or not, but if you've been exposed, and if you are exhibiting symptoms, then to stay home. And we have a culture where we think it's great and noble, and I've been guilty of this, of going to work even if we're sneezing and coughing, because that's just, we're married to our work quite often, and we feel that that's the thing we need to do. But we're seeing the consequences of that, right? We're seeing a pandemic that's not stopping. And certainly in Regina, where is currently the worst outbreak in Canada, and it's over 90% variant. So Regina is the hot spot. And what public health is finding is that in almost all of those cases, what's perpetuating the outbreak is symptomatic people not staying home. So they're still going to visit friends, and they're still going to work. Okay, so the final, or the key piece of how we stop the pandemic with vaccines is that we want to reach this goal of herd immunity. And you may have heard this term, and maybe yourselves be very familiar with what this term means, but herd immunity is the idea that as long as there's enough people who are either immunized or have been infected and have protective antibodies, or just all those green people are not good hosts for the virus, that by having a sufficient number of now immune people in the population, that will start to protect those who either cannot be immunized or who have been immunized, but their immune systems aren't able to mount a fully protective defense against the virus. So herd immunity is key, because there will always be immunocompromised and vulnerable individuals in our communities, and this is how we protect them collectively. And this is also how we drive a pandemic or a virus to extinction. That if we achieve herd immunity, we can actually we can make the virus go extinct. Now, one thing that hasn't been talked much about yet, but this will start to develop over the course of the year, is that to achieve now what we estimate to be about 80% or higher rate of vaccination across communities, that's going to need to include vaccinating children and youth as well. And there's certainly many clinical trials ongoing at the moment to what I think I can safely say is confirm that the vaccines are going to be very safe in children and youth as well. Okay, so which vaccines are currently available? Canada, or the Public Health Agency of Canada has currently, they use the term authorized. So they've approved 4 different vaccines for use. And the vast majority of people who have received a vaccine in Canada so far have received the top 2. And those top 2 are these mRNA vaccines, which I'll talk a little bit about in the next slide. And now there's 2 other vaccines that have just very recently been approved. The bottom 2 there, the AstraZeneca, which is become very famous in the news, and the Johnson & Johnson, which we haven't seen in Canada yet, but it is now approved. Those 2 mRNA vaccines at the top were much celebrated when they came out, and should be, because they're a fantastic success. They are very, very effective. So receiving them is a great way to reduce most chance of severe disease and death. One shortcoming of them has been distribution. So they require a pretty dedicated cold chain. For example, that Pfizer vaccine, when it's produced, say, in a lab in Belgium, it needs to be shipped to a small community in British Columbia. It needs to be stored at -70 degrees for that whole trip around half the way around the world, and then to get shipped over to that community, and then thawed, and then it's ready for injection, but it doesn't last for very long either at that point. So one great development in these new vaccines that have been just rolled out, these ones that are based on these adenovirus vectors, so that's the AstraZeneca and the Johnson & Johnson, is they come with multiple advantages. You'll see that their effectiveness is lower, but I'm going to talk in a couple slides about when not, that's an important consideration. But some of the huge advantages of these next 2 is that they can be produced at much, much higher numbers, and there's more production capacity around the world to produce these adenovirus vaccines, and they are much more stable. So they're easier to transport. They're stable at 4 degrees, so in our refrigerator, for 6 months at least. So you can imagine just how great an improvement that makes for disseminating and distributing these vaccines around a massive country like Canada, and especially around the world, because this has to be a global response to stop the pandemic. Yeah, and I'll talk a little bit about the RNA, getting some questions here. Great, so, and you'll see what the prescribed doses are, and I've got a slide too where I'm going to talk a little bit about the tweaking and the experimentation that's happening right now in the deployment of these vaccines. So first, what's happened in British Columbia so far. So this is the most recent data available as of this morning from Public Health Agency of Canada. And in British Columbia, this is looking at people who have received at least 1 dose, and it does include people who've received 2 doses, of which there's about 80,000 people in BC who have received 2 doses now. The bulk of the vaccines that have been rolled out have been these RNA vaccines. Now, we're not hearing much about these RNA vaccines anymore because AstraZeneca has taken over the news, but you know, through January and February, they were much talked about. And one thing that was said was, or some people's maybe hesitancy around these vaccines, is that it's a totally new and untested technology. It's true that it is a very new technology, and this is the first time delivering vaccines in this way in humans. But it's not really that new. It's been in development for over 30 years now, and it's based on decades and decades of careful molecular biology to understand how these work. And now the proof is showing off just how great they are. So it really, never have we developed a vaccine so quickly against a new pathogen. So you can see how highly adaptable, how rapidly deployed these vaccines can be. Production levels are still a challenge, but that's because they're new. We will get better at producing more of them faster. What's important to recognize is that they're non-infectious, they're non-transmissible. So this RNA molecule that is the vaccine is made out of exactly the same building blocks as all the RNA molecules that are in every cell in your body. And when it's injected in your arm, it's essentially just taking the information to your cell on how your cell can make the spike protein. And so it's teaching your cell the information to make just one little piece of the SARS-CoV-2 virus. And then your cell will make that protein and present it to the immune system, and the immune system can now generate antibodies and develop other forms of immunity. We tend to talk a lot about antibodies, but there's other layers of immunity that are also being stimulated by these vaccines. And that's good, because that means there's lots of layers of backup and extra protection that we're getting. Now, this new one, the AstraZeneca, and you can see that it's starting to be rolled out now. It gets also called Covishield, is maybe what you'll hear if you are offered it. It's the same vaccine, it's just that Covishield is being produced in India under license from AstraZeneca. So it is the same vaccine, it's just marketing and naming issues at this point. So this is a more established vaccine technology. We've had vaccines based on this type of technology for a while longer. It's very adaptable and versatile. It's also based on RNA. So it's bringing that messenger RNA molecule to your cells to teach your cells how to make that spike protein. The difference is that in the AstraZeneca and the Johnson & Johnson, that there's an actual virus that's being grown in the lab to carry that messenger RNA molecule to your cells. That can actually have advantages for how our immune system responds to it. So we might see over time additional advantages that come from this type of vaccine development. But at this point, it's only speculative because we're still so early in the deployment. But as I said, these can be produced in much larger quantities and they're much more transportable. Right, and as I said, both of them are doing exactly the same thing. And I will share these slides afterwards as well, if you want these for your own reference and to be able to look at and scribble on and follow up with me on. Okay, so one thing, BC led the way in this, and it has now been adopted by other provinces and also various nations around the world. And that is changing the prescribed dosage of the vaccines. So when the vaccine trials happen, and a clinical trial is usually a few thousand people to, there's some bigger ones happening now, 30,000 people, where scientists are able to make estimates as to how effective the vaccine is at protecting people from severe disease and death. We can't yet calculate how well it protects from people being able to transmit the virus, but we can calculate how well it protects against disease and death. And in those clinical trials, so the key thing is to make sure that the vaccines are ultimately safe before you roll them out to inject them in millions and millions of arms. And it's long been known that by using a second dose of a vaccine, we are taking advantage of a standard behavior by our immune system. So in nature, when our immune system encounters an infectious disease, it responds in many different ways. And one of the best ways is to make antibodies against that infection. But it takes time, it takes days for your body to find the right antibodies and then to grow them to high quantities. And then once your body has fought off that infection, what it naturally does is it turns down how much antibody it's producing. And that's essentially your body saying, well, you know, I might just not ever encounter that infectious disease again. So there's no point in investing all this energy in making lots of antibodies. And that's just a natural thing that our immune system does. With vaccines, humans are leveraging what our body will do though if it gets re-exposed. So if you get that booster or a second shot, now your body responds as thinking, uh-oh, I'm going to keep encountering this infectious disease. So now I'm going to make high levels of antibody and I'm going to keep them very sustained for long periods. And so that's what that second dose does. Now, you can imagine when the company set out to do these clinical trials, they didn't really know what was going to be the ideal length of time. So that will still be hotly debated as to why the 4 weeks was chosen. But what BC has done and led the way, and this was also partly led by scientists epidemiologists at the BC Center for Disease Control, was they were able to calculate from that early clinical data that in fact, after one dose, 2 weeks after a first dose, people were getting up to 92, 93% effective protection against severe disease and death from just that single dose. Now, when you have a problem that the pandemic is ravaging so far and wide and vaccines are limited, the epidemiologists and the public health officials decided, okay, it's better to get more vaccine to more people faster and we'll delay that second dose until we can come back around and deliver it later, because we will ultimately save far more lives if we get more people that effective first dose sooner. And that's what BC and other provinces have followed suit. And on the right-hand side there, you can see that the implications and consequences of vaccination is different depending on which age group a person falls into. So that's because the virus does different things depending on which age group and what risks. So deploying vaccines to older individuals, 65 and above, which has been the priority across Canada, that's going to save more lives, because individuals in those older age groups are much more likely to suffer severe disease and death. And the numbers are really shocking in Canada. So of people 80 years and older, 25% of those who contracted the virus died from the virus in Canada. So vaccination is key to stopping that. And the great news is that Canada has reached really high vaccination rates now for people in the greatest risk groups, which is in care homes and other group living facilities. Now, vaccinating younger age groups, this is key. It will ultimately save lives as well, but that's what's key to stopping the pandemic, because the younger age groups are those that are spreading the disease more. They're the more mobile, they're the more likely to go out and socialize and have contact with more people. And we do see that more transmission is happening in those younger groups. Yeah, I'll come back and address some of these questions. Just a few more slides here. Yeah, just 3 more slides, and then we'll get into questions. And because AstraZeneca has been in the news so much these past 2 weeks, I thought it would be valuable to take a couple of slides here to talk about how we understand the safety of AstraZeneca. And you can see that heading I've put at the top there, that vaccines are the safest and most effective medicines ever. And that is so true. There's not been any more life-saving thing that humans have ever created than vaccines. And they are so incredibly safe. Their safety outpaces every other medicine all put together. They are so incredibly safe. So how do we figure out how safe they are? So I already described clinical trials where there's a few thousand people tracked. And with the AstraZeneca, some of the first indication of its safety was from taking the 4 clinical trials that happened initially to test its safety. And by sort of grouping those clinical trials together to take all the data, that meant there was 24,000 participants, about half of whom received the vaccine and half of whom received essentially a placebo. So they received either salt water or another completely unrelated type of vaccine. And those individuals were tracked for 3.5 months to see, okay, are there any adverse effects from receiving the vaccine? And the results, so when they tracked serious adverse events, and serious adverse events is a big catch-all, that can be everything from a migraine to a stroke, to a heart attack, to death. One of those 24,000 people did die in those 3.5 months. That person was actually in the negative control group. People die, especially when you track 24,000 people, they die from other causes than receiving a vaccine for sure. So what that result showed is that the number of serious adverse events was actually higher in the negative control groups. So the vaccine is just equally as safe as living everyday life or receiving salt water injected into your arm. So that's the clinical trial. And they confirmed that AstraZeneca is very safe. Now, there's been a follow-up, which I'm going to describe here, and this just came out last week, that the company responsible conducted a major clinical trial across the United States and included individuals in Chile and Peru as well. So this was over 32,000 participants, and they were followed for 3 months to see how they responded to receiving the vaccine. And being such a large trial, they were able to include multiple ethnicities and all adult age groups. And this wasn't broken down evenly. So in this clinical trial, over 21,000 received the vaccine, and the other 10,000 plus received the placebo or non-vaccine. And what they found from analyzing that was how effective the vaccine was. So this boosted their effectiveness or efficacy calculations, and I'll come back and define those terms in the discussion afterwards. But most importantly, what they were really shouting out last week was that there was no evidence of increase in risk of blood clots from that clinical trial. Now, the best way to find out how safe a vaccine is, or dangerous, I mean, this is why we track them, because there can be adverse effects. There have been to other vaccines in history, right? And the question is then, how risky are they and what types of adverse effects happen? But it's really as we start to roll vaccines out into millions and millions of people is when we really see how well do they work and what types of adverse effects are we going to detect. And this data is now about 2 weeks old, but still very important that with the first 5 million people to receive the AstraZeneca, there was about 30 individuals who were noted to have some type of blood clotting adverse effect, right? And this made the news and it caused several nations to actually stop using AstraZeneca for about a week or longer, and also tailor which age groups were being used in. Now, doing the math though, which the epidemiologists can do and excellent statisticians who look at this, they recognize that if you track 5 million people over that same period of time, actually the rate of similar blood clotting events is anticipated to be about 100 events on average each week. So that's just what we notice in normal everyday life. So in fact, there was no increased incidence in those receiving the vaccine. And one important thing to note, why did the blood clotting make the news? Well, it could be an adverse effect that is stimulated by the vaccine. The data doesn't bear that out yet, but we still need to keep watching. But it makes the news because there's really no other time that people spend so much time paying attention to their own health and reporting how they're feeling. And also their medical health professionals are tracking them as well, right? So literally tracking millions of people now intensely to see how are they responding. So we pick up on all sorts of background rates of other sicknesses that people experience. So that blood clotting below the background rate. And so currently there is no evidence that AstraZeneca causes increased blood clots. And so it is at this point very safe to receive. Okay, so I'm just going to wrap up with my last slide here to talk a little bit about some of the research that we're doing. And this is a genomic age. And for the 9 years that I've been running a research lab at the University of Regina, I've been a geneticist that whole time. And I've been using DNA sequencing to understand evolution of microbes and understand microbe-host interactions. But in about 2014, I started to work with public health labs because we could all see the potential of DNA sequencing to help us identify and track infectious diseases. And boy, were we right? This was great that public health labs across Canada were seeing the potential 6-7 years ago to start to get DNA sequencing as part of their routine diagnostics in labs. Now it's taking a long time, but we're now getting there and it's really accelerating because the only way to truly identify variants of concern of SARS-CoV-2, to identify new viruses, identify the next coronavirus, is through genome sequencing. And so it's this genomic age and it's time to increase our ability to deploy these types of technologies in public health. And that's exactly what 2 of our COVID projects are about. So our largest COVID project at the moment is asking the question, what happens when a person is infected with COVID and another infectious disease? Now, first principles suggests that if a person contracts COVID and I put the flu, the seasonal influenza there at the same time, those are 2 really bad viruses. They infect very similar tissues and some of the same tissues, that disease state is going to be terrible and we would expect an increased chance of death, but we don't know. So the best way to survey and figure out what other infections do people have is to use these genomic technologies that we're developing. Normally, one thing to bear in mind is that public health labs do test for other infectious diseases, right? They're doing surveillance, they're testing for flu every winter, but they really only test for about 25 known and top candidate pathogens each winter. And of those 25, about 5 of those are different varieties of influenza and some other common culprits. That there's an immense diversity of infectious diseases circulating in the Canadian population and around the world that we just don't know about because we just don't know to look for them. We can't detect what we don't test for. But then SARS-CoV-2 also, or COVID, created another whole problem this last year, is that testing capacity was not prepared for the amount of infected individuals. And that meant that public health labs had to stop testing for a lot of those other infectious diseases. So now genomics really sells itself because the technology we're developing along with our colleagues in public health is to be able to test for all possible infections at the same time in the same sample. And that's what I've illustrated there, is that a person can be infected with things we don't know that are already resident maybe in their respiratory tract. Maybe we have some indication that they are infected with something else. We don't know what other pathogens are circulating. And another COVID project we have ongoing at the moment is to use this technology to survey bat samples as well. So we're pulling essentially the genetic material of these different viruses out of human samples. I'll show that here. So we get a swab from a person's upper respiratory tract, or a swab from the rectum of a bat. That's where you get most coronavirus in a bat. It's an intestinal infection in bats. It's not a respiratory infection. And then we extract all the genetic material. We convert it to DNA in the lab because that's easier to work with. And then the big innovation is to, what's illustrated there is to isolate the genetic material from the viruses and the bacteria. Because on that swab you see on the left-hand side, most of the DNA on there and RNA is coming from human cells. And the second most abundant source is all the resident bacteria that are natural and very healthy to live in your respiratory tract. The pathogens, the disease-causing agents, are a tiny, tiny, tiny little needle in a haystack. So what we're working on is ways to extract their genetic material, enrich it, and then sequence it. And one of the beautiful ways that we're approaching to extract it is in a very unbiased way. We don't actually need to know what those pathogens look like. And this is what gives us this ability to discover whole new infectious diseases. And I can report that I don't have any data here to show you, mostly because I wanted to cover the other aspects of the pandemic. But what I can tell you is that this is working brilliantly in the bat samples. So we've got novel coronaviruses now where we've been able to pull out their genomes and start to understand coronavirus diversity, which is very important for understanding how do these viruses jump to humans. And one thing that this COVID pandemic has revealed to us is that up until 2 years ago, most betting microbiologists and epidemiologists all had their money on influenza causing the next pandemic that was as destructive as we're seeing COVID. And then all along came a coronavirus that is causing way more damage in the modern era than we've ever seen from an influenza. And this is leading us to now reevaluate past pandemics that we know about in history. And it turns out that some of the bad flus that killed millions of people back in the 19th century actually have more hallmarks of a coronavirus now. So it's quite possible that coronaviruses have been doing this for millennia in human populations. And indeed, we have coronaviruses that circulate as common colds. One thought now is that probably 100 years ago, those common cold coronaviruses were the equivalent of what SARS-CoV-2 or COVID is to us today, and caused millions of deaths at the time. But then eventually, they, over 100 years of evolution, just became like a common cold, and we still see them circulating. So that's going well. And now I have to, this evening, start to write a new grant to increase how much sequencing we're doing to track more of these co-infections, because we found where the technology is lacking, and now we need a bunch more money to improve it. And so that's going to be done, again, in direct partnership with Public Health Agency of Canada, because they're really keen to deploy this in Winnipeg, and then our partners in BC, CDC, Manitoba Health, and Saskatchewan Health as well. And so with that, I have to acknowledge all the different funding bodies that make this possible, particularly those top three. They're funding most of the coronavirus and COVID work in my lab, and those other funding agencies contribute as well through supporting students and postdocs and getting us equipment. Okay, so I talked a bit longer than I had hoped, but I hope that was good and comprehensive. And now we can move to questions. I will stop sharing. And for questions, I can just start going through the chat and tackle those first, and then maybe we'll move to hands if that's, unless one of the moderators has an alternate instruction for me. Emily, is that good? I just go through the questions, the chat?
Yep, that works fine.
Great. Okay, so starting at the top, only one shot needed. So yeah, why don't I just skim back to that slide and I can address questions while having slides up here. One shot needed. I don't believe there is such a thing. I think the Johnson & Johnson sales pitch of one shot is more a consequence of a company bringing a vaccine to market in a very competitive market late. When you see that efficacy, and I'll define that in a second, that's an upcoming question, that's low, the 66%, and there could be higher. I mean, what we're seeing with more trials now is that the numbers are being revised up, which is fantastic. And sorry, what I didn't stress is that those numbers aren't particularly important for individuals under 65 in low risk categories. Everybody just needs to get whatever vaccine comes first, no question. I would also say the same applies for older age groups, but we'll come back around to that as well. The one dose, I think we don't actually know yet what are the consequences of a booster. And in fact, the 4-week second dose is highly unusual. And that's not normally how we do boosters with other vaccines. And that was a consequence more of trying to envision rolling out vaccines to billions of people, making sure you get people back for their second vaccine. All sorts of things we still don't quite know why the companies picked 4 weeks, probably so they could get their stuff to market very quickly as well. So 4 weeks is actually on the shorter side for a vaccine, and it doesn't technically qualify as a booster because it doesn't cause the same response from your immune system. So that's my long-winded way of saying that I think the Johnson & Johnson will benefit from having a second shot. And one thing that's happening now is clinical trials about mixing and matching them, right? Because they're all based on the same protein. They're all using the same spike protein from the SARS-CoV-2 virus. And that suggests that an individual should be able to receive one company's vaccine and then several weeks or months later be able to receive another one of the vaccines. We don't quite know. This is all untested territory, but theoretically that should be possible. Whenever my turn comes up, I will happily go for 2 different ones and mix and match, because there can be benefits from actually having those slightly different vaccine technologies, because they interact with your system a little bit differently and can have different benefits to how they stimulate immunity. In terms of the efficacy versus effectiveness, that's just a technical term in terms of efficacy is calculated when people in a clinical trial are vaccinated. You can't actually tell if they've been exposed to the virus, right? You just have to calculate based on how much virus is circulating in their communities, what are the chances that they became exposed. And that's why you have people who you track who also didn't receive the vaccine, and then you can compare the 2 numbers. And the efficacy is calculated based on the difference between those who received the vaccine and those who didn't, how many people got infected who didn't receive it, and how many people got infected who did receive it. Sorry, I should say show severe symptoms. And that's the efficacy calculation. Effectiveness is, in theory, you could use them really interchangeably, and I think it's fine too at this point to use them interchangeably. Effectiveness is more the concept, or it's used technically to refer to if a person has been vaccinated and they are exposed, how effective was the vaccine at protecting them from that exposure? That's much, much harder to calculate. And of course, we don't do experiments like that on humans of deliberately exposing them. Well, there are some actually experiments like that happening now where individuals have signed up and agreed to be exposed to the virus. So it depends on your pathogen and how many undergrads you can recruit to do things like that. So that has been done in the past. Undergrads have been great for helping us figure out infectious doses with other diseases. I'm sort of smiling because, of course, that would only be done experimentally if it was deemed to be very, very safe. Okay, where's my my chat window there? And sorry, I need to open the participants here so you can put up your hand as well. What does mRNA stand for? I see that's been answered, messenger RNA. So RNA molecules are highly abundant and a core component of every cell in your body. They come in multiple forms, and the messenger RNA form of RNA is the one that takes the essentially just the linear sequence of a protein that needs to be made, and it is the intermediate between the DNA that stores the gene. The gene gets converted, or transcribed is the technical term, into a messenger RNA molecule, and then that messenger RNA molecule carries those instructions to the ribosome, which is the large machine that then reads the messenger RNA molecule and converts it into a protein. And that process is happening in all your cells all the time, all day long. What is happening with these vaccines is your cells are acquiring a new gene they never had before, which is the gene that encodes the spike protein, or a large part of the spike protein from SARS-CoV-2, so that now they can read it. That messenger RNA molecule goes right to their ribosomes, those big machines that convert that sequence into protein. They make the spike protein and then present that to the immune system, so that the immune system can now find the appropriate antibodies to respond to it and develop other types of cellular immunity against the virus. So that then when a person is exposed to the real virus in the real world, they've already got their defenses are ready for it and can destroy and block the virus before it causes serious infection. Yeah, great. The CDC, always a great source for infectious disease information. How the two work against the virus. Lynn, if you could clarify the two which you're referring to, maybe the vaccines. I'll let Lynn, if you're still there, to put up your hand and clarify your question for me. Kate, okay, I got the efficacy done. Side effects. Yes, so these vaccines do have notable side effects that people are suffering. Fever, general malaise is quite high. Something like almost 25% of people receiving the Pfizer have some sort of ill feelings soon afterwards. But those all so far turn out to be transient, and it's your body responding to the fact that it's got a high dose. So your body is receiving a lot of a foreign viral protein. That protein can't make you sick, and it can't replicate, and you can't transmit it to somebody else. So that's all very safe, but your body still sees something foreign and is raising its different immune alarms, which is good because that then reinforces its ability to learn and develop those defenses against the infection. Lynn, you can unmute yourself and ask a question or a comment.
Andrew, I think you already answered it. I asked that question, and then in your next couple of slides, you explained how the process works in the body.
Oh, great. Okay. Nancy raising a point that the blood clot risk is in females under 55. I've not seen that specifically addressed by the statisticians I'm following yet, but it's definitely something I will look into. And Nancy, if you wanted to comment or follow up on what you've seen about the increased risk for women for blood clotting.
Hi. Actually, I can't remember where I saw it. It was something that was on social media. It was a video that I recently saw explaining or demystifying the risks. But yeah, that's what I thought it said, was that that women of childbearing age and women that are like the type of blood clot was the kind that women get when they're on oral contraceptives. But I can't cite exactly where it was that I saw it now.
Yeah, and I think, and as I tried to cover, that is certainly a possibility, right? This is a foreign agent that is being injected into your body, and your body's mounting an immune response. And the risk with our immune system is that our immune system is incredibly potent at what it does, and it can cause adverse effects. And that happens just in regular everyday life, right? So we've heard of Guillain-Barré, which is essentially a consequence of your immune system freaking out inappropriately. There's different types of toxic shock. There's all sorts of flesh-eating disease is in fact your own immune system destroying your tissues. It's not the actual pathogen doing it. So our immune systems are fantastic because they keep us alive every day, but they are also dangerous to us because they can be so potent and can result in these adverse effects. And so the blood clotting one is one that will certainly continue to be tracked. One thing I do want to say though is that because the real power in understanding the risks comes from the clinical trials, but even more so is examining population level when millions and millions of people, and millions of people is in different parts of the world. So if ethnicity has any type of determinant on risk factor, that's where we really start to see, because unfortunately what happens at this point when so many people are desperate for the vaccine, a lot of people are very much against the vaccine, that singular events of a person suffering a severe adverse reaction can make global news or even the evening news in Canada and seem like a terrible consequence of receiving the vaccine. But it's important to remember that for many individuals, especially those in the higher risk categories, 65 and older, many were already at great risk of suffering different adverse effects from blood clots and are suffering blood clot related disease in their everyday lives before receiving the vaccine, right? So the risks were already heightened. And then when you consider receiving the vaccine and then track that individual for 3.5 months, there might have been every chance that they were going to develop a blood clot anyway, regardless of having received that vaccine. And so that's where we have to be really careful about taking as much evidence as possible to calculate what are the true risks on a population level of receiving these vaccines.
Mm-hmm. That's what, I guess that's what my concern was, is that I wanted to see the specific data for that particular demographic, you know, women of childbearing age, especially women on oral contraceptives. And I haven't been able to find anything about the AstraZeneca, you know, in that's that specific. It seems as though the data that I've been seeing is for like general populations. And so I was worried that the efficacy or the safety claims were based on general populations, and that particular demographics risk may have been watered down a little bit because they weren't being looked at specifically. But I thought it was that, I thought that was the main reason why some of the countries in Europe were hesitant, and that they approved it and then stopped that briefly. Yeah. And I thought that was the reason, was because of the added increased risk for women of that age. So, yeah. But I can't.
Now, I think one optimistic note is that we learn so much about human health when there are massive vaccine campaigns like this, and so many people will be tracked so carefully that if that risk does exist and is statistically significant, it will be picked up quickly. And that's way better than so many other things that people consume or have injected in their bodies that are either deployed to just a few hundred people around the world or not tracked to the same level. That we really do get such a great understanding about human health and what these vaccines are doing just by virtue of the size of this vaccination campaign globally. A method to clean out our noses or upper respiratory tract. So as in a safe way to try to blow your nose without spreading the virus, I guess, is maybe what that question is about. Oh, and I skipped one there, I'll come back to it. I don't know, I think, again, I would just reiterate that it's one of those things to always be very cognizant about how much are you spreading potential pathogens, either COVID or other things that could be replicating in your respiratory tract. So hand washing, I'm a huge fan of it as a microbiologist, and I don't mean to suggest that I'm in any way paranoid about infectious disease, not at all. I embrace all microbes. I know that I'm only alive because of all the many microbes that live on my body that I do share with other people and acquire from the environment around me. But when it comes to infectious disease, yeah, just being careful about how we exhale and how much hygiene we perform on just keeping our hands and tissues carefully so that we're not putting others at unnecessary risk. So how long is the spike protein being produced after being vaccinated? Is it different between the two vaccine types? Yeah, that's the kind of question I love, because that's the type of research I'm really into. And unfortunately, I don't have an answer for you. But what I can tell you is that when these are still very experimental about how these RNA molecules are being designed to be read and produce the vaccine. And in that 30 years of development before deploying them, what had been learned is that you can, of course, engineer these molecules to last longer than normal messenger RNA. So on average, normal messenger RNA lasts minutes in a cell. So you can do special capping and different design features to make sure that they last longer so cells can produce more. You can also encode other signals to tell your cells to produce extra amounts of that protein. And one thing we don't hear much about is that when Pfizer first went to clinical trials, they were actually trialing 2 vaccines, and one of them had a smaller piece of the spike protein, but it was being produced at higher levels. And they found that it didn't actually work very well because cells seem to be overproducing it and resulting in different complications and problems that wasn't mounting as good an immune response against it. So there's still some experimentation in developing this new technology, but it works great. And Emily, if I find out, I'll certainly come back to you to let you know how long that production continues in those cells. So why is getting the test to see if you had COVID 2 or 3 months back only? How to find out how long the antibodies stay in our system? Yeah, well, that's a $1 million question still, about how long do we keep producing the antibodies, but more importantly, how long does our body produce protective concentrations of the antibodies? So that's one sort of caveat when we look at testing diagnostic techniques, is that they're incredibly sensitive. So people who test positive, for example, for the RNA test, which is the standard nasopharyngeal swab that takes overnight at the public health lab, that can detect down to probably viruses that have already been cleared by the immune system, and we're just detecting fragments that are left over from a cleared infection. With the antibody one as well, being able to detect the antibodies versus what's protective, we don't have a good sense yet. Where we might be getting better ideas is previous studies that looked at the coronaviruses that cause common colds. And in those cases, individuals can be reinfected by those common cold coronaviruses 6 months to a year afterwards. But I don't want you to go home with that number, because there's a whole different population dynamic going on with those cold viruses, because they're so diverse and they circulate constantly in humans, that it might not be so much a feature of a person's immunity declining over those 6 months to a year, by just the fact that they're in fact being exposed to a completely different variant. So that's a dynamic we don't quite understand yet. But it does, I think, illustrate how we have to stop this pandemic, because the longer we let it go and the more the virus diversifies, the harder it is to contain it by mass immunization. So I'm watching for hands, but I guess folks are okay if I just keep scrolling through the chat here. StatsCan sent out COVID questionnaires and tests randomly. Oh, Kate, what kind of test was sent out randomly? Because there's been multiple being trialed at the moment, and I'm curious what one.
Yeah, I was one of the lucky candidates, and I had symptoms in March 2020, because I'd been at a women's march. So of course, stayed home for 2 weeks, and then I had to add another 2 weeks. So anyway, they sent off this random thing, and it's a big, complicated questionnaire, which I filled out, but they wanted you to take your own blood sample, which I can't do and won't do. And I'm in a bubble of one, nobody's coming near me, right? Everybody knows that. So anyway, so I refused to do the blood sample, which if I had, they said they'd let me know what else was going on in my system. I'm not sure I wanted to know. But anyway, so I didn't give them my blood work, but I did send the questionnaire, and I haven't heard anything back. Now that was, oh golly, that would have been, before, in a March, it would have been January, February, March, I can't remember, January, February maybe. But I was interested because it was, sent priority post. It was, and of course, I was the only one I heard that ever received this. So, but yeah, so I you aren't involved in that, obviously.
No, but I did attend a meeting with one of the scientists who's overseeing testing blood samples across Canada to understand immunity and the
Yeah.
antibodies present. And what they're drawing from is mostly Blood Services Canada samples so that they can
Okay.
test hundreds and thousands of people across Canada. And one thing that, now granted, another thing about the infection, right, or the pandemic, is that it's going in these different waves. And some of that data comes from the first wave and very early in the second wave. But one thing I thought was particularly interesting that they had noted was that by looking at antibodies, so essentially deducing who had been exposed to the virus, whether they knew at all that they were infected, most people didn't, is that the outbreak dynamics from that way of tracking were very different in Quebec and Ontario compared to Saskatchewan and Alberta. And in Quebec and Ontario, the outbreak was much more limited to very specific groups of individuals, whereas it was maybe a more democratic virus in the prairies where it was spreading much more widely across the adult population. And I mean, from that, of course, we can make hypotheses to look at differences in behaviors and approaches to controlling the pandemic in those different provinces. But that early evidence suggested that actually the outbreaks had quite different dynamics depending on which province of Canada you were in. So there's been lots more people exposed really in Manitoba, Saskatchewan, Alberta by November than in Ontario, Quebec, even though mortality rates or more people had died in Quebec and Ontario by that point.
Can I just add, my nephew and niece in Ohio, in the States, both got it, different symptoms. I wasn't very happy about this because of my older sister. But anyway, they had different symptoms. They recovered. She was in college. He was going to work. Not then, he had to stay home. But anyway, he ended up giving his plasma, is that right? So they could study to see what, is that, okay. And that was in the States. And well, you know what's going on there. Better now, but still. But do they do that here, take plasma?
Well, certainly Blood Services Canada is still involved in that.
Okay.
Because that's always a part of your blood. And that's the bit where the antibodies are essentially concentrated.
Okay.
It's the non-red blood cell part of your blood. Yeah, so that's still being monitored. But again, that's not as great a surveillance as we would like to understand population level infection exposure, because it's certainly not truly representative of the Canadian population, those who go to give blood to Canadian Blood Services. So there are other studies ongoing, but they would have smaller sample sizes as well. So there's trade-offs that where you get your samples from. Okay, so here, Rick, a great question about developing vaccines in Canada. Yeah, so Canada, I mean, back in January and February, we were not looking like we were doing very well on vaccine rollout, even though Canada had purchased the most vaccines per capita of anywhere on Earth. We were one of the slower countries. And last time I looked 2 weeks ago, we were still 56th in the world for rate of vaccine rollout. And I don't know where we are now, but I'm going to guess we're probably about the same. And it's because Canada doesn't have a domestic production, or that's one piece of the complication, is that Canada has never invested in that production. Now, Canada, through agencies like NSERC, has done fantastic research to develop vaccines. And there's a Vaccine and Infectious Disease Control Organization in Saskatoon that specializes in developing vaccines for animal pathogens. And they are now receiving tens of millions of dollars from the feds to ramp up production to make vaccines against SARS-CoV-2 for the Canadian population. So we will start to get domestic production thanks to this pandemic, but it's not happening yet. So there's also some in Canada developed vaccines that are in clinical trials now, yet to be seen. But what Canada will start to produce first are things like the AstraZeneca on license from AstraZeneca. It's sort of like brewing Guinness in Canada on license. You get the recipe, you start making it in Canada. So hopefully that will help us both in this pandemic, but certainly help us for future pandemics. So no, I'm not involved directly in vaccine development, but and this is one thing that's really come out just in the past couple of weeks on our work on the bat coronaviruses, is that coronavirus was not a very attractive research topic 2 years ago. So we didn't really know much about its diversity. It's this huge group of viruses, and we knew they were diverse, but we didn't really know how diverse. And what we're learning about the bat coronaviruses now, due to the sequencing we're doing in my lab, is we can start to figure out just how much diversification happens in the spike proteins relative to other components of the virus. And that both tells us how mammalian immune systems fight the virus. So we can really deduce how the virus is spreading and immunity is arising in bats, but it also helps us predict which bat types of virus are going to be best able to jump into humans. And the ultimate goal is that when we understand the structure of spike well and where these different mutations happen that make it able to bind to human tissues, then we can start to develop vaccines against viruses that may be circulating in nature that haven't had the opportunity to jump to humans yet. So we can anticipate future pandemics and stop them before they happen would be the ideal. So in that respect, yes, the type of research we do will eventually help inform vaccine design. Where will we be in 6 to 12 months? Yeah, well, I try to keep a smile on my face and be optimistic about these things, but I'm also a realist around how humans squander opportunities. And when it comes to infectious diseases, we have a long track record of either squandering or just developing a tolerance threshold. And within Canada, we just have to look at various infectious diseases that are already terrible in Canada that the Canadian population just has a tolerance threshold, like tuberculosis, for example, that I think that's where we're headed with SARS-CoV-2, that there's so many barriers to achieving that herd immunity. We have the opportunity, but human behavior and other dynamics are going to get in our way, and I don't think we're going to achieve it. So I think what's going to happen this next winter, in some ways, for some individuals, it could be a worse winter, because one thing that happened this current or past winter is that for the first time ever, we didn't have a seasonal flu season. There was no community transmission of influenza in Canada detected. That's never happened before. Public Health Agency of Canada never met the threshold to actually declare flu season, so it technically just didn't happen. That's probably not going to be the case come fall. There's so many pressures to, of course, open up for many good reasons, right? I know that we have to find the right balances, but I don't think we're going to find the right balance in the safest way. And instead, what's going to happen is that our society is just going to start to tolerate a certain level of a deadly virus circulating in our population in the years to come. Now, vaccination is fantastic to protect those who are willing to get the vaccine, but there are a lot of people who are not willing. And more sadly, there are many who can get the vaccine whose immune systems just aren't capable of developing protective immunity, right? So if you're on any type of immunosuppressant or have an immune dysfunction or cancer patients, there's news out today about looking at delaying the second dose is maybe not a good strategy for those fighting cancer, because their immune systems are already compromised and they need that second dose sooner. So I think in 6 to 12 months, it's going to be a dirty situation, that there's going to be a lot of disease still circulating in Canada and around the world. So yeah, I think the best we can do is to continue to spread the good word about the effectiveness of vaccines. Again, like I said, we've never had a medicine as potent as vaccines. Things like smallpox and polio, right? Those are not challenges we face anymore that used to kill hundreds of thousands of people into the millions each year. And vaccines safely got rid of them. Well, almost got rid of polio now. Measles, I mean, we could eradicate measles, and unfortunately that's a deadly disease that we're just not able to tamp out yet, because there are people who won't get the vaccine. And unfortunately, I think that's what's going to happen with COVID. CRISPR technology can be used to edit genes. Whoa, that's a big one. So I'm going to defer the CRISPR to Jim and Linda. If you would like, I can certainly give a whole other presentation on CRISPR, and I think it really deserves it because it's a very powerful technology. It certainly has huge implications for manipulation of human genetics, animal genetics, the genetics of pathogens. Never have we had a tool before so powerful to be able to go in and edit genes in a human or other living thing. But I think that's about all I should say here, or we'll get totally off the SARS-CoV-2. And Lynn, I'm sorry to hear about the loss of your mother and Guillain-Barré. Yes, so Guillain-Barré, a very unpredictable consequence of really just your body having its defensive barriers breached, right? So Guillain-Barré can be triggered by a scalpel when a person goes for surgery. And the rates are actually, so far appear to be lower rates of Guillain-Barré in people receiving the vaccine than individuals going for surgery. So that's very heartening. Again, that just suggests how safe the vaccine is by comparison. But it's one of those things that just happens because our immune system is there to protect all the inside parts of our body. And if something comes through, and that could be a stick cutting you while you're gardening. Gardening also has risks of triggering your immune system. So thankfully, the vaccines appear to be very, very safe compared to just even general surgery. Great, a note about the PI clinic. Hydrogen peroxide lessen the risk of infection. Well, hydrogen peroxide, using it in a mask, on a mask filter, it would certainly be a powerful decontaminant. It is very good at killing things. I don't know how effective it is against SARS-CoV-2. One nice thing about SARS-CoV-2 is it's actually a really wimpy pathogen. So when it's on a surface, it's dying. Well, I wouldn't technically call it alive, but it's losing its ability to cause infection quite quickly. So its half-life is somewhere around an hour or so, meaning half the viruses on a surface will become non-viable in an hour, and it decays continuously from there. And it's also very easy to decontaminate with hand sanitizers, even just washing it, so basic detergents. So like other respiratory viruses, or it's actually weaker in the external world than a lot of other respiratory pathogens. So that's good. That means that things like simple decontaminants that we might have around the house are a great way of decontaminating surfaces or even washing your mask. I don't think there's a way to really hydrogen peroxide your mask at the same time as wearing it in a safe way, and probably not really necessary. More the best mask hygiene is to wear it when you're out and about and around other individuals, and it's mostly a community protective, right? You're protecting other people from the pathogens that you may be carrying more than you're protecting yourself from inhaling, but it does also reduce the chance of inhaling external viruses and pathogens. But then when you're done at the end of the day, if you've been wearing it for an extended period, definitely wash it because you've been exhaling all sorts of other microbes into it that will happily grow in that moist mask. Which types of masks are best with variants of concern around? Yeah, so that's true. I would have probably answered that question a bit differently a few months ago before the variants were becoming such a problem. So cloth masks, certainly cotton masks are better than synthetic masks. It tends to be how sort of fluffy is the material. So all the small little fibers, the more of them there are, the better they are at catching what you're exhaling. And certainly 3 plies of cotton have been shown to be better than 2, and there's lots of information on different good mask designs. And certainly the recommendation to double mask will prevent even more transmission. If you can get them, the N95 masks are some of the best because they are so good at blocking small particles like viruses. The masks to avoid are those that have any type of valve on them. So those are actually really bad because they don't work as a community protective mask because they're designed to protect against the person inhaling small particles. But then when they exhale, they're actually pressurizing their exhalate to now shoot out more virus, and that could actually distribute the virus even further if an individual is infected and wearing one of those masks that has a little valve on it. So those are best to be avoided because they're counterproductive to what the basic function of the mask is to stop that transmission. Okay, ethical considerations of CRISPR, a fascinating discussion. Well, sure, I mean, if there's no more SARS-CoV-2 and wow, it's hailing out, then I can certainly talk a bit about CRISPR. But anyone else have other comments or questions, follow up on coronaviruses? Big doses of vitamin D. So my understanding is that of all the studies done on vitamin D yet, none have detected significant contribution to helping cure the disease. Canada funded 2 massive studies, each one $5 million last year to look specifically at vitamin D. I haven't seen the outcome yet, but we will get the results very soon from those studies in the Canadian population. So that will be the most pertinent to that discussion because it will have been studied in Canadians. Oh, and CRISPR is a subsequent presentation, sure. Maybe not in April. This is going to be a tough month ahead, but come May, certainly, I'd be happy to do something like that.
Thanks very much. That was a great presentation, followed by lively questions. I think we'll just stop the questions for now because it's already 2:30. But if you have any more in the future, it sounds like Andrew is willing to answer your questions later, so you can send him an email. Rick, did you want to say anything before we sign off?
I do. What a great neighbor to have, Andrew. You just just wander over every day and ask him a new question of the day there. Yeah, thank you, Andrew. That was pretty awesome. And thank you, Bev, for suggesting Andrew as well. Bev thought Andrew would be a pretty topical presenter, so thank you for that. Just a couple of more community announcements for you guys. Whoa! Better hurry before we lose power here. We're hoping to have a Fire Smart public meeting sometime next week. It'll be on fairly short notice, but we're just waiting to hear from the city on rules around burning and that kind of stuff. So we're hoping to share that with everybody. So it'll be a fairly short notice meeting, but we're hoping to do something next week. Then, of course, April 7th is our vaccination day at Beacon House. We'll see how that all turns out. April 10th, any artists who want to come down to Mindy and Kelly's place to have a look at what we can do to restore the mural there, they're more than welcome. And then April 11th, Craig's going to have his book reading. And then May 1st is Fire Smart Day. So you can bring all your stuff down to the bins. That's a Saturday. Have supper, and then Saturday evening or Sunday evening, enjoy the PI Players as they put on their show with the Savannah Sipping Society. So April's going to be a busy month, like Craig, Andrew said. So buckle up. That's it for me.